Hi, welcome to Student Tutorials. In this lesson, we will be talking about the order of draw. So, what is order of draw? It is a specific order to filling evacuated tubes from a syringe or directly into an evacuated system. This has been developed to avoid cross-contamination from one tube to the next. Hence, by following the correct order, the accuracy of test results is guaranteed. To begin with, the first in the order are the blood culture bottles. They are also called the sterile collection bottles. Here is an image. Blood culture bottles are used for microbiology tests which help in determining the presence of microorganisms in the bloodstream which can cause infections such as bacteremia and septicemia. They also come in varying colors of tops. Some have green, some have orange. Aside from that, there are also two types, the aerobic bottles and the anaerobic bottles. And using this too, the aerobic bottle is to be used first, followed by the anaerobic. The reason behind is there is a possibility for air in a syringe which could affect the results. Also, maintaining its sterility is top priority, hence, it should be the first to be collected to avoid contamination. The next on the list are coagulation tubes. They have a light blue top. Here is an image. They are used for hematology tests such as prothrombin time or the PT, active partial thromboplastin time or the APTT, fibrinogen assay, D-dimer, and more. It has an additive of sodium citrate which acts as an anticoagulant by binding to calcium ions in the blood and disrupting the clotting cascade. After collection, it has to be inverted 3 to 4 times. It is followed by the serum tubes. There are two types of serum tubes. The first one has a red top. Here is an image. They are used for chemistry tests and blood bank tests. It commonly has no gel or additive. However, some have silica clot activators which helps the blood clot. The second type has a gold top and commonly known as the serum separating tube or the SST. It is used for biochemistry tests such as the lipid profile, urea electrolytes, protein assays, and more. It has two additives. First, it has silica particles which activate clotting, causing blood cells to clump together. The second is the serum separating gel, which floats as a layer between the blood cells and plasma, forming a physical barrier between them which provides ease at obtaining the serum. After its collection, it has to be inverted 5 times. Next on the list are the heparin tubes. They have green top. Here is an image. They are used for biochemistry tests which requires heparinized plasma or whole blood for analysis. That said, this contains heparin, which acts as an anticoagulant. After collection, it has to be inverted 8 to 10 times. The next one are the EDTA tubes. There are two types of EDTA tubes. The first one has a lavender top. Here's an image. It is generally used for hematology tests where whole blood is required for analysis. Some of these tests include full blood count or FBC, erythrocyte sedimentation rate or ESR, HbA1c, blood film, and more. Moreover, it is also used in blood bank tests, such as cross-matching. It has ethylene diamine tetraacetic acid or the EDTA as an additive. After collection, it has to be inverted 8 to 10 times. The second type of EDTA tubes have a light pink top. Here is an image. It has the same function as the lavender top EDTA tube. However, this is specifically used for whole blood samples being sent to transfusion labs. It also has EDTA as an additive and it has to be inverted 6 to 8 times after collection. The last tube is the fluoride or oxalate tube. It has a gray top. Here is an image. It is used for biochemistry tests such as tests for glucose and lactate. It also has two additives. First is the sodium fluoride. Sodium fluoride acts as an antiglycolytic agent which prevents further glucose breakdown after the sample has been taken. The second additive is potassium oxalate, which acts as an anticoagulant. After collection, it has to be inverted 6 to 8 times. Now that has been the order of draw. Now if you want to memorize it, I have another tip. Here is an acronym I use to remember the order of draw. BC Chef. B stands for blood culture bottles, C is for coagulation tube, S is for serum tube, H is for heparin tube, E is for EDTA tube, and F is for fluoride oxalate tube. Now if you're wondering why the order of draw is like that, blood culture bottles have to be collected first to prevent bacterial contamination of the blood culture samples. The coagulation tube is collected next to avoid any additive from other tubes getting into the tube. Heparin tube should be collected before the EDTA tube because EDTA would alter chemistry tests on the heparin tube. And since heparin doesn't affect EDTA, it should be collected next, followed by the fluoride oxalate since EDTA has no effect on testing from the oxalate tube. That would be all. Now this has been Student Tutorials, and if you find this lesson helpful, hit the like and subscribe buttons, and click the bell button to get notified for future tutorials. Thank you.